Okay. Good morning, good afternoon, and good evening. Now that's the new routine we have to say to cover the entire globe. Welcome to today's IJMF Spotlight Seminar. We have Arezoo, but I'm not going to do the introduction. That honor goes to Pavlos. Pavlos, take it from here and please introduce our distinguished speaker and we can get going.
Great, thank you, Bala. And thank you all for joining and attending this seminar. This is truly a great pleasure and an honor to introduce my colleague, Arezoo Ardekani. Arezoo actually is in mechanical engineering at Purdue University, and was, before COVID, now we're all working from home, my next door neighbor. So every time I had a question, I would just stick my head in her office and have a great discussion with her about a wide range of topics. As you may have read in her short bio, Arezoo has actually been distinguished in many different ways. I'm not going to cover everything. I will start by saying something that it's not in her bio, that it is one of the best colleagues and collaborators one can have. And I've been very fortunate for that. But going into some of her accomplishments, Arezoo has been the recipient of NSF Career and the NSF Presidential Career Award, the PKs, has been recognized for her outstanding work as a graduate student mentor and an early career award from our College of Engineering as for excellence in research, and many, many other awards across numerous other societies, including many distinctions from invited publications and journals. She's a prolific scholar. She has published over 100, I think 110 journal papers, all in high impact, highly recognized journals in our field. And her research actually has covered a wide range of applications and topics. With that, I will stop the introduction and pass on the floor to Arezoo for her current talk. Unfortunately, I cannot attend to the duration of the talk. I will have to jump out, but Bala will moderate the discussion afterwards. Arezoo, thank you for doing this, and the floor is yours.
Thank you, Pablo, for the nice introduction. And I indeed cherish our collaborations over the last few years. Let me share my screen. Are you able to see the screen?
Yes, Arezoo.
Yes,
very
we
well.
can.
Great. So I'm going to talk about hydrodynamics-mediated trapping of microbes. But before I get started, I would like to acknowledge the role of those students and postdocs who have contributed to what I'm going to present today. Most of what I'm going to present today had been done by Nikhil Desai and Wasim Sheikh, who are students in my group and are now postdocs at École Polytechnique France and University of British Columbia, as well as by Rishabh Moore and Rajat Dandekar, and Gaojun Li, who was a student in my group and is now assistant professor at Shanghai Jiao Tong University. So the motivation for what I'm going to present is from the Deepwater Horizon oil spill that happened in 2010, and 4 million barrels of crude oil was released in the Gulf of Mexico. And microbes were our first respondents. And if those microbial blooms were not right after the oil spill, the catastrophe would have been far worse. What I'm going to talk about is that how hydrodynamic interaction between those oil droplets as well as soluble components of oils with those microbes play a role. In fact, in the aftermath of oil spill, 7 million liters of dispersants were also used with the hope of reducing the impact of the oil spill and increasing bioremediation by reducing the size of the droplets and increasing the surface area. So this schematic from our cover image of the paper shows some of the catastrophe, some aspects of the catastrophe. And here, what I'm going to talk about is that how hydrodynamic interaction between these microbes and liquid-liquid interfaces and air-liquid interfaces play a role. So if you think about life at low Reynolds number, most microorganisms are very small, and consequently inertial effects can be neglected compared to their viscous effects. And there are also a number of other processes that affect their life and dynamics, including the external background flow. They live in turbulent environment and their background flows. They're also living in the world that has patchiness and gradients in nutrient that drives their dynamics and distribution. When they get close to surfaces or interfaces, they form biofilm. And if you think about some of the plankton that have larger sizes on the scale of centimeter, and then inertial effect becomes important, we cannot neglect it any longer. So I'm going to touch on some of the aspects that we are showing in this cartoon. These microbes, for example, bacteria, can have single or multiple flagella that are attached to their cell body through a motor. This motor has a stator and rotor. And if you look at it, it looks actually very much like a mechanical motor. For bacteria, that these motors can reverse its direction of motion. And for bacteria that have multiple flagella, that can lead, like E. coli, that can lead to formation of bundle of flagella. And once in a while, these motors reverses its rotation and leads to tumbling. And we'll talk about some of those aspects later on in the talk. The life at low Reynolds number was studied in a seminal work by Parcell. And I didn't know actually Parcell did his undergrad at Purdue before I moved to Purdue. And later he moved to Harvard to do his PhD in physics, and he received his Nobel in physics for his work on NMR. So what he states is that time doesn't matter, and the pattern of motion is the same, whether slow or fast, whether forward or backward in time. And this is referred to as the scallop theorem. If you think about a scallop, it moves in water because it can open and close its shell at different rates. And because of the inertial effect that time irreversibility in motion leads to its propulsion. But if you put it in corn syrup or very viscous medium, as shown in this video by Taylor, even though this flapper, which is a mimic of a scallop, can move in water, it cannot propel itself in very viscous medium. And microbes, of course, know this rule of physics, and they break symmetry somehow, for example, by having helical flagella and being able to move forward, but because there's a fore-aft asymmetry, and it leads to their propulsion. So because the governing equations are Stokes and linear, then one way to look at a motion of these microbes is to look at superposition of point force singularities that are solution to the Stokes equations. If you think about settling a sphere, a far-field solution of a settling sphere can be modeled as a point force singularity. For a swimming organism, the thrust is generated by the flagella and drag acting on its body, and this equal and opposite force can be modeled with a force dipole. Their finite size can be captured by a source dipole, and asymmetric propulsion can be captured by a force quadrupole. If you think about a helical flagella that is rotating, then this equal and opposite torque that is acting to the flagella and cell body can be captured using Droplet dipole. Superposition of all of these point force singularities, which each have their own flow field, can be used to look at different physics for these swimming microbes. So I'll start by a simplified model of a microbe interacting with surfactant-laden droplet to understand the role of hydrodynamics on this interaction between microbes and surfactant-laden droplets. So droplets here for this portion of the talk is modeled as a sphere, and bacteria is modeled as a force dipole, where dipole strength is dependent on the thrust that is generated by bacteria. Force dipole is a good model for swimming E. coli. For example, this experiment by Goldstein and co-workers show that the flow field is very well captured by force dipole. Now, if we have the image singularity that is needed to satisfy all the boundary condition on the surface of this drop, then we would be able to calculate the hydrodynamics-induced velocity and angular velocity. And from that, knowing the swimming velocity of the microbe, we would be able to update its position as well as orientation, here shown with P. But the solution for such a problem was absent in literature. So first, we needed to find the solution for a point force singularity outside of surfactant-laden droplet. So this work was done by Vassin, where basically we find the image singularity that is needed to satisfy all the boundary conditions, including zero normal velocity on the surface, tangential continuity of tangential velocity. And we also have a jump in bulk shear stress that is balanced by Maragoni stress and interfacial shear stress. Then for these surfactants, they follow surfactant transport equation when there is convection and diffusion of these surfactants. And in general, surfactants can be transported into the interface and out of it. But for the purpose of what we are presenting, we assume we have insoluble surfactants. I won't go through the detail of the derivation for this point for singularity. It's a long derivation, but the detail is given in these papers if you are interested. What we will do here is use those solutions to be able to understand trajectory and dynamics of these swimmers. So in order to do that, we use Faxon law. And from Faxon law, and knowing that these swimmers are force-free and torque-free, we can find the hydrodynamic induced velocity and angular velocity at the location of the swimmer, which depends on the flow field caused by these image singularities. And then the cross product of this stress generated by the swimmer dipole strength and the strain rate also gives us the angular velocity, and the aspect ratio plays a role here as well. So let's see how the results would look like if we have a swimmer or a microbe near a droplet. For small droplets, these microbes get scattered, whereas if you have a large droplet, it gets trapped around the droplet. And this is all caused by hydrodynamics interaction between these droplets and microbes. In fact, if you look at the phase space, distance versus angle around the swimmer, distance of the swimmer from the surface of the droplet and its orientation, we see no fixed points if droplets are small. But for larger droplets than a critical trapping radius, we see a saddle point. So in subspace, some region in the phase space, we have microbes that trap around the droplet, and in the rest of the domain, they would escape. So this critical trapping radius depends on one parameter that is very important, and it's dipole strength. The dipole strength is the thrust generated by the bacteria by the flagella, normalized by the viscous force. And this trapping radius reduces with inverse of dipole strength square. And it gets smaller and smaller as we go from bubble to oil to rigid sphere as we increase viscosity ratio, here shown with lambda. And this is the case for a clean droplet. So as we increase viscosity ratio, we have a stronger and stronger hydrodynamic interaction. But if you look at the surfactant-laden droplet, we see that this critical radius goes down for surfactants because the hydrodynamic interaction becomes stronger, but it's even below the case of a rigid sphere. And this is non-intuitive to have surfactant-laden interface that has more hydrodynamic interaction than rigid sphere, but we'll talk about where it comes from. And this is consistent with the results we get for the trapping time. So if we calculate probability distribution function for the trapping time, we see it's the lowest for the bubble. Then for oil droplet, it gets larger. If we add surfactant, it gets even larger. But for surfactant-laden bubble, this trapping time is the largest. So the factor viscosity ratio is basically opposite when we are thinking about clean droplets versus surfactant-laden droplets. So this is coming from the fact that the flow field around spherical surfaces can be written as a sum of surface solenoid field and surface irrotational flow. So if you have a droplet that has interfacial viscosity of 0, then this flow field is written in terms of surface solenoid field plus solid particle, which is surface irrotational field. The first one is the same as clean droplet. So now if we have surfactant and non-zero interfacial viscosity, for the case that point force singularity is normal to the surface solenoid field disappears, and the solution is the same as solid particle. So what this does is that when we increase viscosity ratio for clean droplet, basically normal velocity, tangential velocity, and angular velocity of the microbe all increase, and that leads to less trapping. Whereas as we increase viscosity ratio for surfactant-laden droplet, we see that normal velocity is unchanged, but tangential velocity increases and angular velocity goes down. And this leads to less trapping. So that's the reason the effect is opposite. So this role of surfactant is also shown here when we look at now distribution of these bacteria, but including also rotary diffusion because of the run and tumble. So for cases when droplet size is above the critical trapping radius, We see some of the microbes trapped, some escape. That's because of the rotary diffusion, which leads to escape of microbes. But as we add surfactant, we see most of them are trapped near the droplets, and surfactants increases the trapping time. We wanted to test these effects experimentally, so we did experiments for Pseudomonas putida, which is a motile bacteria degrading hydrocarbons, as well as looking at E. coli. So the setup is like we have bacteria in suspension of bacteria in culture media. The bottom is confined by a cover slip, and on the top it's confined by a liquid-liquid or air or gas-liquid interface. And we look at distribution of these microorganisms or bacteria. This is also relevant to other microorganisms like Tetrahymena, that is a microorganism living at air-water interface, as well as Pseudomonas aeruginosa, which is a bacteria causing lung disease and also colonizes at the interface. So the way these experiments are done is using inverted microscope and visualizing bacteria at different plane away from these solid surface as well as gas liquid surface. So these are different planes shown at different proximity to these interfaces. And we count number of bacteria, and from that we can get probability distribution function for these bacterial distribution. So you see here there's a peak near the solid surface, and this had been reported in the past for bacteria near solid surface. But here what we see is that near gas-liquid interface, we have a larger peak in the distribution of bacteria. This is CO2 liquid interface. For air-liquid interface, we don't basically report the data because we do have larger peak as well, but the reason for that is taxis toward more oxygen. Whereas for CO2, we don't have such effect. So everything we see here is due to hydrodynamics effect. So this is non-intuitive results, but consistent with what I described earlier from our theoretical prediction. So we looked at a number of different interfaces, CO2 interface, dodecaine, mineral oil, soybean, and for all of these, we basically look at probability distribution function for bacteria. For larger viscosity ratios above 1, we see that these different PDFs follow the same behavior. And they are the same as what we predict from our theory. Basically, what the theory shows is also when we have viscosity ratio that is large, the rotation rate of bacteria near interface is independent of viscosity ratio. Because we include other singularities like droplet, we are able to also capture this rotation or circular motion of bacteria near interface as well. And the theory and experiments follow the same behavior. We later used this theory to look at thin films and look at distribution of microbes in thin films that are confined with liquid-liquid interface on the bottom and air-liquid interface on the top. So what we see that suspensions of pullers are accumulated near interface. Pullers generated thrust in front of their body. And this accumulation is stronger than suspension of pushers that generate thrust behind their body. And what is interesting, we see that short flagellated bacteria are accumulated near air-liquid interface, whereas long flagellated bacteria accumulate near liquid-liquid interface. We have not tested this experimentally, but this is doable because we have bacteria in wide range of geometries, long and short flagella, and that can be tested. What I discussed here was looking at hydrodynamics effect, but bacteria also show chemotaxis. They sense and have direct movement in response to chemical gradient. So for example, for E. coli, they can have multiple flagella. And when the motors are rotating in one direction, they form flagella bundle. They move straight, but once in a while, one of these motors rotates the other direction. They unbundle and go to a different trajectory, different orientation. And the runtime and tumble time depends on the concentration gradients for the nutrients. So the phenomena that I discussed so far, hydrodynamics, is a short range effect in the sense that it's valid only a few body length away from the droplet, whereas chemotaxis can bring bacteria from far away to the nutrient source. So if we add chemotaxis on top of hydrodynamics, we see, of course, a stronger chemotaxis leads to more accumulation near the drops. But what is interesting is that still the role of hydrodynamics, which is shown here with dipole strength, is very strong. So this is shown here by trapping time, normalized by the simulation time. So this trapping fraction basically significantly increases as we increase dipole strength. So even though chemotaxis is essential in governing the interaction between microbes and droplets, but hydrodynamics role cannot be neglected. And this becomes relevant also when we look at soluble components of oil as well. So this acts as nutrient distribution around these oil droplets. And if we calculate the average nutrient exposure around these droplets, we see that non-chemotactic bacteria are not much affected, but the ones that are chemotactic can have much more increase in their average nutrient exposure. So basically, the average concentration that they're exposed to during their travel near nutrient source. So this is because chemotaxis brings them close to the droplets, and when they get close to the basin of attraction of hydrodynamics, then hydrodynamics kicks in, and then it leads to the trapping near the droplets. We already touched on soluble component of the oil. For example, methane is an example of that. So the question is now how motile and non-motile bacteria consume these soluble components. Is it at different rate? Does chemotaxis help? And how hydrodynamic interaction affects these processes? So in order to look at that, we look at these rising motion of droplets in a field that has some patchiness in the distribution of methane or any other soluble component of the oil. And then we also include chemotaxis of these bacteria, so they swim toward region of higher concentration. The questions we want to answer as they are interacting with these rising motion of oil droplets is that does chemotaxis benefit motile bacteria over non-motile ones? What factors dictate this benefit, if there is any, and what is the extent of this benefit? So we solved this problem computationally, looking at solving for continuity equation and conservation of momentum. Here we have surface tension force to capture the interface of droplets, and this is done using front tracking method. And so we solve for a convection diffusion equation for methane, and the sink term here are coming from bioremediation caused by motile bacteria, shown with M, and non-motile bacteria, shown with NM. Then we need to solve for a convection diffusion equation for a distribution of these bacteria. For motile bacteria, we have the chemotaxis bias. They're swimming around with velocity Vs, and they can be some diffusion present because of the run and tumble effect. And for non-motile bacteria, they basically act as a flow tracer. And then these bacteria can reorient. They are not spherical, so we need to include their aspect ratio. And their reorientation rate depends on their rotation due to ambient vorticity caused by these pseudo-turbulence because of rising motion of these droplets, and also because of this their chemotactic reorientation, which is related to this gradient of concentration. So here is a region of high concentration methane. This patch of high concentration methane will get mixed as this rising motion of droplets occurs. And but there are still some regions of high gradients in concentration of methane, and that drives patchiness and distribution of motile bacteria. And we see the same places where we see large concentration gradients, large consumption rate by motile bacteria, but that's not the case much for non-motile bacteria because they act as flow tracer and their trajectory and dynamics is not governed by chemotaxis. We define motility benefit, which is the consumption rate by motile bacteria and non-motile bacteria. We can get difference of this consumption rate. And we look at some of these parameters that affect this motility benefit. I highlight only a couple here in this talk. One is droplet size to see how that affects the motility benefit. We see that as we make the droplets smaller, we have larger motility benefit because we are looking at a constant volume fraction. So a smaller droplet corresponds to larger volume fraction. And that leads to creating regions of sharper gradients, and the distances that are traversed by these motile microorganisms to reach their nutrient-rich environment gets shorter, and that's why we see larger motility benefits. And this distance is also shown here. We see it increases with time because we get more and more mixing in time, and consequently they need to travel longer distance to get to these sharp gradients in concentration. Another parameter that is important is swimming speed, because the distribution of these bacteria depends on their swimming speed, and the faster they go, they have also a stronger chemotaxis effect. So motility benefit linearly increases with this swimming speed. And the reorientation rate that this bacteria have to sense this gradient of concentration also leads to increase in motility benefit, but it saturates, it doesn't linearly increase with motility, with reorientation rate. For the next few slides, I'm going to switch gears and talk about different hydrodynamic interaction and different physics that lead to hydrodynamic interaction of these microbes with the surrounding environment in oceans and lakes. And that's governed by density gradients. Density gradients in oceans and lakes can be caused by either variation in temperature or salinity. And these regions of sharp density gradient are referred to as pycnoclines. It has been reported that large concentration of marine snow particles or planktonic organisms are found at these pycnoclines. Even some species of krill or dinoflagellates cannot swim through thermoclines or holoclines. Even at this larger scale, if you think about planktons, their bloom formation is correlated with seasonal stratification. And these phytoplankton bloom, phytoplankton consume CO2. So when they die, they sink to the bottom of the ocean. So it's a natural way of carbon sequestration, and it leads to, it's called biological pump. But they can be also toxic and lead to disruption of water supply system. And as we have enhanced in stratification because of the climate change, more and more form. This image is from Barun and Darling in Australia, but more recently there were algal bloom forming in Lake Erie near Toledo and disrupted water supply. So if you think about these organisms, bacteria, plankton, even larger organisms like zooplankton, the question is that how their length scale compare against a stratification length scale. There's one length scale in the problem, which is the length scale over which density changes, and that's on the order of meters. So if you look at that length scale, we may say, okay, they're not much affected. But what we have shown is that there is another length scale related to viscosity, diffusivity, strength of stratification, and gravitational acceleration. And using that length scale, even organism with length of about 600 micrometer will be affected by a stratification. So in order to understand the fact, we started with the same procedure that I talked about earlier, looking at point for singularities, but now looking at how a stratification modifies these point for singularities. For a settling sphere, we have a point for singularity, and we see stratification generates these toroidal eddies. And stratification suppresses vertical motion of fluids. So if you think about qualitatively similar, this is similar to a point for singularity between two walls. And in that case, we also see these toroidal eddies. For a forced dipole, we also see these toroidal eddies that form. And this forced dipole would be a far-field solution for a bacteria. The velocity field also decays more rapidly in a stratified fluid. This is velocity caused by stratification normalized by the one in homogeneous fluid and decays rapidly. And this can affect the mechanosensing because many of organisms, plankton and zooplankton, detect a predator or prey using mechanosensing, like this copepod has this large antenna, and deflection of this antenna can detect flow disturbances in the fluid. So if we quantify this detection volume of these organisms as the volume inside which flow disturbances exceed the threshold, we see that it scales with the size of organism over this stratification length scale to the power -3. So it's relevant to Rayleigh number. So if you think about 600 micrometer prey, then its detection volume is reduced by 50%. So predator-prey interaction can be affected. But these organisms not only also mix fluid, not only they overcome viscous dissipation to swim, but also mix a fluid around them. And consequently, their energy that is expended by these swimming organisms increases in a stratified fluid. And that scales very similarly with Rayleigh number as well. So these are experiments showing different organisms with different pixels, like zooplankton and other organisms that are homogeneously distributed in homogeneous fluid. But for salty fluid, where fresh water is added to the top, they go to the interface. And this is not because of salt gradient, it's because of density gradient. The author actually added more sugar to the top to get uniform density, but it's still having salt gradient, and the distribution goes back to homogeneous distribution. We looked at Heterosigma cashewa, which is algae causing red tide in salty water, and added fresh water, and they accumulated near the interface as well. So these are relevant, especially in the context of climate change, because stratification has enhanced because of the climate change, and that leads to reducing the portion of oxygen circulating below mixing layer. So we have larger abundance of microorganisms above in the mixed layer, but below that, we get less and less oxygen because of this enhanced stratification. So ocean is losing its breath. Some regions have lost 40% of oxygen. Volume of water containing zero oxygen has more than quadrupled, but there are different reasons causing it. One is solubility, because water gets warmer and it holds less dissolved oxygen. The stratification is another reason that I talked about. And the metabolic rate of these organisms also increases by 30 to 50% when the temperature goes up by 2 to 3%. So that leads to generating large-scale ocean hypoxia. So we hear a lot about global warming, but if we think about climate change, we need to talk about all three pillars: ocean acidification, ocean deoxygenation, and global warming all together to understand the entire process. So if you want to look at such a problem, though, it has very rich physics. So in order to explain this physics, let me start with a simpler problem first, and then we add the fact of stratification. So let me start with the Eocene problem that all of us studying fluid dynamics are familiar with. So if we want to look at the drag of a sphere or an object in a viscous medium, but include the effect of inertia, then we see that we have a length scale, which is called Eocene length scale, which is 1/, Reynolds number. And inside this length scale, close to the surface, close to the object, viscous effects are dominant. But as we go outside the Eocene length scale, inertial effect balances viscous effects. And so we need to consider this length scale when we study the enhanced drag or modified drag of object in weakly inertial effect. And now in the case of stratification, we have an additional length scale, which I talked about, this is stratification length scale. So very close to the object, we have viscous effects that are dominant. Outside the stratification length scale, viscous effects balances buoyancy effect. And outside the Eocene length scale, we have inertia balancing viscous effects. So this is stratification length scale, is the length scale of the buoyancy effect when Peclet number is small. When Peclet number is large, it actually changes and finds another form, which is size of the object over Richardson number to the power 1/3. And Richardson number itself is a dimensionless parameter that is the ratio of buoyancy to viscous effects. So now, similar to Eocene problem, we need to use singular perturbation method to understand how a stratification modifies the dynamics of objects or swimmers in stratified fluids. So we use a singular perturbation method where this parameter epsilon is the length of the object over stratification length scale. So in the inner zone, we have only viscous effects that are important. And in the outer zone, we add this point for singularity and to solve for the problem. But then we need to basically match the inner solution and outer solution and find the role of stratification. So there are three scenarios that we need to separately understand and discuss. One is when buoyancy balances viscous effects and density is governed by diffusion, when buoyancy balances viscosity and density is governed by advection, and when inertia balances viscous effects, and that's when stratification length scale is larger than Eocene length scale. So once we look at each of these scenarios separately and rescale the variables based on the small parameters we have and neglecting nonlinear effects and solve for the singular perturbation method, we can actually find the enhanced drag. So if you have a rigid sphere, this is how the enhanced drag looked like. The parameter F is actually long, so I'm not reporting it here. But for droplets also behaves very similarly, except that we have this term R, which is the role of viscosity ratio. You saw actually exact same term when earlier we were talking about interaction of microbes with droplets, which captures the role of viscosity ratio. So the solution would be the same as bubble when viscosity ratio is 0, and the same as rigid sphere when it's infinity. This enhanced drag again follows the same three behaviors. If you think about the case where this stratification length scale over Oseen length scale is very large. That's when inertia balances viscous effects, and this stratification enhanced drag actually becomes proportional to Reynolds number, the same as Oseen length scale. But this solution that we are talking about here is valid when we have weak stratification. If we have strong stratification, we need to look at the problem numerically. This is a sphere in a stratified fluid, which generates this jet behind the sphere in a stratified fluid, whereas in homogeneous fluid, we have downward flow behind this sphere. In a stratified fluid, we see even formation of these Lee waves behind these spheres. Now, stratification leads to some interesting effect also when we look at interacting spheres. For example, for two spheres in tandem, we see drafting, kissing, and tumbling in homogeneous fluid, but in a stratified fluid, we see tumbling is suppressed. We call it drafting, kissing, separation. For homogeneous fluid particles in side by side, we see them moving away from each other because the vorticity is blocked by the presence of the other sphere. But in a stratified fluid, we see even though initially they move away from each other, eventually they move closer and move as a single object. Now, looking at suspension of particles or drops, this slide is related to drops. We see similarly stratification affects their hydrodynamic interaction in the sense that their rising motion, these are lighter than background fluid, is slowed down, but also as we increase the stratification or reduce fruit number, which is the ratio of inertial force to buoyancy force, we see they are not only slowed down, but also less dispersed and more clustered. Their cluster formation can be also analyzed using pair correlation function, which I talk about in the next slide. But for now, if we look at the Reynolds number of single droplet and suspension of droplets, we see stratification slows them down. This stratification has a stronger effect for suspension of drops compared to single drops, and velocity fluctuations are significantly reduced when we add the stratification. The microstructure of suspension is affected, and if you look at the probability pair probability distribution function, we see there's peak for nearly touching droplets, but the peak gets larger as we increase stratification. And looking at the angular distribution, also there's a peak for 90-degree corresponding to horizontal clustering, which forms in suspension of drops, but the peak gets larger as we increase the stratification. So stratification leads to more accumulation of droplets and particles and enhanced clustering. Density stratification, when we look at objects like spheres or cylinders or microorganisms, they can also mix fluid around them. So for example, here we are looking at a single sphere in a stratified fluid. Here is a suspension of 28 spheres, and here is 56. We see very clearly the wake behind these spheres. We see these volume of fluid that is basically trapped around these spheres and goes from region of low density to high density. And this oscillation is caused basically because of the stratification itself is correlated to Brunt-Väisälä frequency, which is the frequency of a fluid element being displaced from its neutrally buoyant level. So this drift is a mechanism leading to mixing for these suspensions. But when we look at cylinders, the change in potential energy that is caused by settling of cylinders is more governed by the vortex shedding that occurs for cylinders, whereas we have less of that for spheres. I would like to go back talking about the swimming dynamics that has been the theme of the talk, but understand how stratification can modify swimming dynamics. So for this, we use a model swimmer called the swimmer, which is a model for ciliates. Some of the ciliates are shown here in this figure where their cilia are covering the surface of these organisms, and the mitochondrial beating of cilia leads to their propulsion. We want to see how stratification modifies their swimming dynamics. So this swimmer model basically assumes we have a slip velocity on the surface of the swimmer. And by playing with this slip velocity, we can get basically pullers that generate thrust in front of the cell and pushers that generate thrust behind the cells. the flow field generated by pushers and pullers are shown here, where pullers are basically bringing fluid from front and back and expels it from the side, and pushers do the opposite. This model had been shown in and used widely in the literature to look at either swimming speed, nutrient uptake, interaction of the swimmers, mixing by the swimmers, as well as looking at the effect of inertia and stratification, which is what I'm going to talk about here. So what we see is that these pullers and pushers in weakly stratified fluid or in inertial regime behave very differently. The pullers in inertial regime or weakly stratified fluid becomes unstable, whereas pushers are stable. And as we add the strength of stratification, we see exactly opposite behavior. The reason for this is that if you look at the puller that is moving on a straight line in a weakly stratified fluid, it's bringing fluid from front and back and then expels it from the side. So if you look at the inertial effect and having a memory of basically the flow field from previous time step, you would see that this flow field that it generates pushes it away from this straight line and consequently becomes unstable. Whereas for pusher, that is generating the flow that is coming from side and expels it from front and back, it brings back the swimmer to the straight line. So it becomes stable. Now, if you think about the effect of stratification, it is exactly opposite in the sense that the flow field caused by the stratification and deflection of isopycnols, it generates the opposite flow field and makes the pullers stable when we look at the strong stratification. And this is what we are seeing in these videos as well. But as impact on mixing that is caused by swimmers as well, and I'll talk about it in the next slide. But here I showed the phase space. In this phase space, on the top region of the plot, we are looking at pushers. In the bottom, we are looking at pullers. And this color code is showing the strength of stratification as we go from the outer circle, which is for homogeneous fluid, to inner circle. We look at the strong stratification. And on the vertical line, we're looking at very small inertial effect. On the horizontal line, very large inertial effect. So in the outer ring, we have very strong, very weak stratification. And for pullers, we have unstable behavior, which we saw in the previous slide. And for pusher, we have unstable behavior for very strong stratification. This affects mixing of these swimmers. For weakly stratified fluid, for example, we see these pullers undergoing helical trajectory and pushers undergoing more straight trajectory, so consequently causing more mixing. This is also clear when we look at two swimmers interacting. This is for swimmers in weakly stratified fluid inertial regime, where we see them undergoing this circular regime for pullers and undergoing more straight trajectories when we look at pushers. So this has consequences for life of these organisms. These were the dimensionless parameters we discussed that plays their important role. For pullers, we saw them for weak stratification going basically straight, whereas for pushers, the same thing happens for weakly stratified fluid, and the behavior is opposite. When we look at the strong stratification, we see pushers become unstable, and that can lead to trapping of these at phytoplankton layers. So the questions that we answered here is that combined effect of inertia and stratification, how they affect the swimming speed and stability. So stratification stabilizes inertial pullers while it destabilizes inertial pushers. We talked about effect of stratification on swimmer interactions and showed that stratification can increase contact time, and talking about mechanisms behind the accumulation of organisms in horizontal layers, which can be the fact that these change in stability causing their trapping and leading to different dynamics and circular motion when we look at colliding swimmers. For the next couple of slides, I talk about some of the other work that we have done that are related to the theme of today's talk, which is trapping of microbes and hydrodynamics effects. We looked at how flow affects distribution of microorganisms. And in these, for example, microfluidic devices, these vertical flow that are present can lead to patchiness in distribution of microbes and even leading to formation of biofilm streamers. We look at how fluid elasticity or non-Newtonian nature of the background fluid interact with these swimmers, and in the presence of flow that leads to emergence of limit cycle and lead to patchiness to their distribution and concentration of microorganisms. We look at the sperm, showing that in Poisson flow, they migrate towards center line because of hydrodynamics effect. In extensional flow, they can undergo buckling, and the computation results and experiments capture the same thing in terms of buckling dynamics. Looking at microbes in complex fluid, we basically look at how the hydrodynamic interaction affects their distribution near surfaces, and how rheological properties of fluids like shear thinning or viscoelasticity can affect their interaction with surfaces. And the shear thinning itself, for example, leads to very small viscosity close to the swimmer itself. So the swimmers move very rapidly in corridors of low viscosity medium. And with that, I'm going to summarize the talk. So the first part of the talk I summarized by a limerick. Thanks to hydrodynamic interaction, surfactant help to increase bacteria fraction, consuming oil droplets, eating them like chocolates as it's combined to chemotaxis reaction. And the second part of the talk, I talked about mechanosensing and how the speed and mechanosensing can be suppressed by stratification, and stratification suppressing dispersion of particles and enhancing their cluster formation, and the mixing that is generated by these swimmers being affected by their stability in inertial regime as well as a stratified regime. And with that, I'm going to stop and open the floor for questions.
Thank you very much, Arezoo. Good tour of a lot of wonderful things you have been doing. The floor is open for questions. Please unmute yourself and feel free to ask questions. Shankar, you have a question?
Yes, Arezoo, very nice talk. I had a question. How big is the length scale of those clusters compared to, say, a gradient of the mean concentrations?
You're referring to the first portion of talk where we have concentration in, for example, soluble components of the oil. Is that correct?
No, I was thinking about the stratification problem.
Stratification.
And right.
So the length scale of cluster is several particle size, whereas the constant, which would be on the order of millimeter and centimeter, the length scale over which density changes is on the order of meters.
Okay.
Though I should mention that I'm studying these at the smaller scale. The computational tools I'm using basically is fully resolving different length scales. So consequently, I'm not looking at the scale of meter.
I got it. Okay. So you think there may be an opportunity where they may be even bigger? Is that not clear at this point?
It's not clear for me at this point, but there is opportunity that they can be bigger. We need to study it, basically.
Sure. Very nice talk. Thank you so much. Yeah.
Thank you.
Can I follow up on that question? So in the real ocean context, the salinity gradient, is it, and the thermal gradients, are they large enough for stratification to affect these micro scale organisms?
Yes, that's actually what we showed. In, like, one length scale that comes to mind very immediately is that the length scale of meter and the stratification being length, but being weak. But the calculation that I've done is for the gradients that occurs in for example, Pacific Ocean. That was taken from a paper that measured density gradient. So for it, those numbers also occur in ocean, and that leads to stratification length scale that can be on the order of few hundred micrometer or millimeter. So still smaller organisms can be affected by stratification. Of course, if you think about bacteria or something that is 1 micron, then won't be with this mechanism. But there can be other mechanisms even for those smaller bacteria. For example, what we have shown in a study that I didn't talk about here is that for some smaller bacteria or algae, that are below this stratification length scale, but they show phytoplankton. They show, bioconvection plumes because of the bottom heaviness. So stratification, what it can do is suppress these bioconvection plume formation because the bioconvection plume itself has a larger length scale. So stratification can still suppress those bioconvection plumes.
Ed, do you see any questions on YouTube channel?
Oh, I don't.
Okay. Floor is open for other questions. Anyone wants to Jing Ran, you have Alfredo, go ahead.
Yeah, Arezoo, thanks a lot for the nice talk. I think that when we have thermal stratification, there is an extra shear which is introduced in the flow field, and the extra shear can actually let the microorganism lose their orientation. So somehow, is this the main mechanism, or the scales are too different?
I think one should look at both of them coupled together, and the scales are not that different. So in terms of the scale of shear, the driving some patchiness because of the gradients in velocity field, as well as these patchiness we see in concentration of either salt or temperature. So I think the scales are not different, and one has to look at them and couple with each other.
Thanks, yes.
Any other questions? Otherwise, I will be asking one more question. So I will first give you guys an opportunity. Okay, Arezoo, it's my turn again then.
Sure.
I want to go to the first part of your talk where you talked about trapping, very interesting results. And you have not just interesting results, a very innovative analysis looking at this multiple aspects and making use of it. The one question I have there is clearly the microbes are small, so Stochasian approximation works well. But these droplets that trap them could be maybe millimeters or hundreds of microns in size, and they could have inertial effects. How do you, I mean, have you taken that into account? But it may be harder from the multipole expansion perspective. So how do you handle that? That's my question.
Mm-hmm. Yeah, that's a very good point. So for the first part of the talk, we use the multiple expansion for microbes, not for the drops, but still the point you are making is valid in the sense that the droplet themselves, if they're rising or settling, inertia effect can change their motion. We looked at moving droplets as well, but still neglecting their inertia. But the second portion of the talk that I talked about, that was numerical simulation. So there we included the inertial effect, the rising motion of the droplet, and even the pseudoturbulence that it generates. So there we included all those effects into the problem.
Yeah, okay, so it's a difficult problem. But it's interesting that I always thought that even for finite Reynolds number, so in Euler-Lagrange kind of simulations, we only apply the force. There's always an interest, like force coupling method of March and Maxey includes higher order terms. So the systematically introducing Rotlet and other things into the two-way coupling, what effect does it have is something that I've been interesting. So I need to read your papers on the trapping effects. Okay.
Yeah, that's actually a very good point. Like I've been also interested to use some of those approach. I haven't get a chance to look at it, and now you're reminding me of that. This would be an interesting direction to look at as well.
I have another question, Arezoo. Can I, Bala?
Please.
Yeah. So once these small bacteria trapped at the interface, do you think that there might be an effect on surface tension, so they might act like surfactant?
I think they will be. So here on the experiments, actually, we see some of them also trapped at the interface, though we primarily look at that their distribution away from the interface. But theoretically, we are just modeling the surfactant, not the bacteria or colloids, modeling bacteria as colloids and then including their effect, but that can well happen as well.
Shankar, you have a question? Please go ahead.
Yeah, thank you. Thank you, Balaya. Also, so my question is, if you look at this in the natural environment, I'm thinking of, and I'm very naive in this, so forgive me if I'm thinking of these red algae blooms that you see in Florida and stuff like that, very large scale distribution of microorganisms. Do these things occur on such large scales, and what are the simulation techniques to model that kind of a thing? And does your work kind of feed into that?
The simulation techniques to look at those red tides would be not to definitely like resolve the microbes like very last slide we did. There is no way to do that, but some models similar to what I presented for the consumption of methane, for example, where you model these microbes as convection diffusion type equation, but then you need to include all the physics that are relevant there is doable. And the paper that I was referring earlier in response to Bala's question, where we looked at bioconvection plume and how they are suppressed, actually was with the goal of that. So there we solve basically continuity and momentum equation, but rather than, but the microbes themselves are continuum scale, and then include the Chemotaxis effect and some of the other effects. And we see actually what you're referring is that the stratification basically suppresses bioconvection plume, leading to accumulation of microbe at the surface. So some type of calculation with those type of analysis, which includes other physics that are needed for the red tide formation, would be relevant.
So it is a multi-scale problem. And my question is, do the physics at the micro scale, like right at the scale that you're looking at, do they actually affect things at the large scale? I suppose that's
I believe so. Yes, it does. Okay. Because and the problem also becomes more challenging, because if you think about the multi-scale is not only physical multi-scale, but also biological multi-scale, because the smaller organisms and driving also larger organisms and all those scale become relevant in that sense as well.
Yeah. Thanks. Yeah.
Okay. Any more questions to Arezoo? Okay, Arezoo, I think it's about 1 hour. So thank you for spending your time and going through the many facets of the research your group has been doing. And thank you for this wonderful talk. And we all will meet in 2 weeks' time with yet another Spotlight seminar. Thank you very much.
Thank you very much.